and an honor to be part of this symposium. My lab designs and uses models to decode how genomes work, how they evolve, and how they break in disease. And I have the privilege of directing an institute in San Francisco based on the idea that we can build better models, but also better research technologies in the wet lab if we're doing these two things, these two types of technology development hand in hand. Rather than doing what I've done a lot of my career is just wait till the next machine comes out and then try to build methods to analyze data from that machine. And so today I want to talk about sequence to X models. So X could be like the activity that we heard about from Anshul. It could be the protein folding into the protein function that we heard about earlier today. Sequence to some kind of thing that we care about. Neural network models that can do that. The idea of using them hand in hand with very precise genome editing, not just the kind of perturb-seq screens we heard about from Aviv this morning, but being able to go in and put these rare variants that Stephen was just talking about individually into an isogenic background and test their effects. So genome editing is getting very good, and it's an amazing technique in all of its different formats. But there are so many possible experiments, as Aviv really beautifully highlighted this morning. And this can roadblock us in terms of what experiments should we do. And often we end up doing random ones or screens rather than sort of smartly designed experiments. And so the premise for our work is that by building models that are reasonably accurate, they don't need to be perfect, but good predictors of the outcome of experiments, that we can design better experiments and then use the data from those experiments to design better models. So I'm going to briefly tell you about a couple of arenas in which we're doing this at the Institute, and then I'm going to delve into one of them in more detail. The first one is developing better immune cell therapies. And in particular, trying to get things like engineered T cells to work on solid tumors, which is a really important clinical objective and very difficult at this time. And so here we're using machine learning in two ways. One is related a little bit to the gene PT that we heard about from James earlier, learning from single cell and spatial transcriptomics data, the relationships between genes and proteins and how they work together in tissue, and how certain cells might be functionally different depending on where they are in that tissue, like an immune cell right in the middle of the tumor versus an immune cell further away. And learning those patterns, then we can knock genes out, genes that are defining these cell states, knock them out or overexpress them in cells and assay them through live imaging. The ML comes in again in terms of data acquisition from the live cell imaging, and really rapidly try to develop more effective therapies. The second example is in addressing antibiotic resistance. And here we're trying to use nature's bacteria killers, phage. And this is already being done many places where people, when someone has an antibiotic resistant infection, go and try to screen lots of phage that have been collected from out in the world, in ponds and in soil and in other patients, and see if any of the phage might be able to kill their bacteria. But this is very inefficient and is not scalable. Our idea is that if we could learn the rules of what sequences in a phage make it be able to kill a genome, given the sequences in the bacterial genome, if we really could extract those design rules, we could build phage from scratch. They're small, you can just synthesize them. So can we synthesize phage from scratch by learning the design rules? And that way you could quickly, with a patient who's dying from a bad infection that's not responding to antibiotics, much more quickly reboot a phage and put it in and kill their infection. The third example, which is what I'm going to spend the rest of the talk on today, is establishing causality for genetic diseases. And we've heard a lot about this throughout the day. Our idea is to build these sort of sequence to activity or sequence to X models hand in hand with folks who are developing very precise genome editing. Something called an editron, where we can do base editing at any kind of bases, not just certain kinds of bases, and that we can edit not just one nucleotide at a time, but replace an entire haplotype. So thousands of base pairs of DNA. And the tool from Seth Shipman's lab called an editron does that by coupling the CRISPR-Cas system with something called a retron, whose job in bacteria is to synthesize DNA. So we're using this is a template to repair the genome with the sequence that we designed to put into it. That gives us, on an otherwise isogenic background, a way to test the effect of these high-scoring variants from the models, and then we're reading them out at single cell and single molecule level, including with the SAMOSA assay from Vijay Ramani's lab. So let me show you what that looks like in terms of, I just showed it as sort of a technology stack, but it's actually a cycle. This is like the lab in the loop that Aviv also spoke about this morning. The idea is we start with the experimental data and the technologies that exist today, use those to build a sequence to activity model, use the model, turn it around with the techniques for variant attribution that we've been hearing about to score variants, prioritize them, understand them, think about them, and use the results of millions of computer experiments where we say, well, what if I change this variant and that variant together, all the different combinations, develop causal hypotheses, and then go back in the lab, test those, generate data with this high throughput genome editing platform, and then that data will help us build the next generation of models, which will hopefully be much better than the okay models that we have today. So we've been applying this at the levels of genes and proteins. I'll speak a little bit today about gene regulation and gene expression. But before that, I want to talk about what's going on in this picture, which is to make a nice protein and to make an RNA, the regulatory sequences that control expression of genes need to physically get nearby the genes that they regulate through genome folding. And so I'm going to talk about a sequence to genome folding model, and then we'll go on and we'll talk a little bit about gene regulation and making personalized predictions for that. So I'm going to take you around the cycle for a modeling approach focused on the folding of the genome. The data that we can start with is called chromatin capture, or Hi-C. It does not measure an actual folded structure of a chromosome, but instead assays with sequencing, proximity ligation, and then paired DNA sequencing, the frequency with which pieces of DNA across a genomic locus are close to each other. And it looks like this, a heat map on the right. Both axes, the horizontal and the vertical one, are genomic coordinates for the same piece of DNA. The diagonal is the polymer itself, and then the off diagonal is measuring how frequently pieces of DNA that are far away on the linear polymer are nearby in 3D space. So it's a 2D representation of a 3D structure that's easy to measure through sequencing, so we can get it at scale. The redder the pixel is, the more frequent the interaction compared to what would be expected by chance, given how far away they are, and blue means it's less frequently interacting than expected by chance. So every piece of the genome has a picture that looks like this. They vary a little bit across cell types and tissues. They vary a lot from one locus to another. And so our idea was to build a model called Akita that takes a megabase, 1 million base pairs of DNA as input, uses a convolutional neural network with a 2D output head to predict the map for that sequence with no other data inputs. There are models that perform the same task using epigenetic data or open chromatin data, and they are a little bit more accurate. But the beauty of this model is that it has sequence only as an input. So we will be able to turn it around and ask questions about perturbing the sequence without needing to make any additional measurements. So fitting this neural network involved feeding in lots of paired examples of a target of real data, and then trying to tune the parameters of the model so that the predictions look like the targets. We do that in a classic situation with a training set, a validation set to help us with hyperparameters and to avoid overfitting, and then a completely held out test set of genomic regions. And we achieved a very low mean squared error, genome-wide 0.14, which essentially means the predictions look like the pictures, what you see here. This is a typical concordance. So with a model that's that good, not perfect, but pretty good, we can then turn around, because it's a sequence-only model, a model that takes only sequence as input, we can say, well, what happens if I change the sequence? So right now, if I have a sequence that predicts this picture, it's quite easy on the computer to edit the sequence. Genome editing is getting more high throughput in the lab, but it's really high throughput on the computer. So I can make deletions, inversions, tandem duplications. I can make point mutations. I can do all kinds of combinations, and I can do millions of those in one day. So the model will take each of these altered sequences and make a prediction. And sometimes they don't look very different, and other times there are notable differences. There's several in this picture, but I'm highlighting here, because I'll come back to it later in the talk, this increase in contacts adjacent to deleting these base pairs. Now, we can see that with our eyes. To do this in an automated way over millions and millions of variants and lots of 1 megabase windows across the genome, we need statistics. We have a paper coming out soon in a bioarchive that's been up for a while talking about how there's lots of ways to quantify whether that map looks different from that one. And they're not the same. So depending on what you ask for, you get different high scoring variants back. But we were able to sort of summarize which statistics capture which kinds of features. So now we can apply this at scale. Here, I'm applying it to a cohort of families, healthy parents, a healthy sibling, and a child with autism spectrum disorder and/or developmental delay from the Safari simplex families. And then we score every de novo variant, every variant that's present in the sick child, but not in their other family members. And then we ask the model, do these disrupt genome folding or not? And so a higher log disruption score means more disruption. And what you see here at a cohort level is strong evidence that while many variants in both the sick kids and the healthy siblings do not disrupt genome folding, there's, as evidenced by the higher red bars compared to blue bars on the right-hand side of the plot, there's more genome disruption caused by the variants in the sick kids than by the variants in their healthy siblings. We saw the same thing in another large cohort of whole genome sequences from families with children with a congenital heart defect. And so, while this doesn't prove in any individual patient what Stephen was talking about, being able to clinically execute on a specific variant and say that it's causal, it does show that at a cohort level that this mechanism of disrupted genome folding could play a role in both of these developmental disorders, which is super important, because only less than half of these children have a disruptive protein coding variant or splice variant, a variant that's clinically actionable or could explain their disease right now. In fact, in autism, it's only 20% of the kids who have something that's a likely causal variant. And so there's a huge need to be able to understand for the families what might be making their child sick, and a huge need scientifically to say, are there maybe other pathways or causal mechanisms that we're not thinking about right now for these diseases? So in looking at those high scoring variants, we were able to develop a hypothesis, which is about enhancer hijacking. What that looks like is here now I'm just drawing a linear genome and it's a cartoon. Suppose there are some regulatory sequences, enhancers, that could, in theory, turn on this gene on the right-hand side, but their chromatin interactions are going off to the left-hand side of the slide there. And the reason for that is that there's a chromatin domain that's folding over there, and there's one that's folding over here, and they're insulated by this boundary element that's has the protein CTCF sitting on it. And this is a known role for the protein CTCF to create these insulating boundaries. Now, suppose the sequence underlying that CTCF binding site, not a sequence in the protein, not a sequence in a splice site, not a sequencing variant that disrupts an enhancer. All the pieces are intact, but the variant is knocking out the binding of CTCF. Now those enhancers can come over and turn this gene on at the wrong time or place during development. And you can imagine the opposite, and we saw evidence for the opposite as well, where chromatin interactions get disrupted by a variant. So they can both create a new interaction and destroy one that should be there. Now, this is a testable hypothesis. We can go into human cells, induced pluripotent stem cells, to test this hypothesis. We can do this in a sort of low throughput way right now with traditional CRISPR genome editing, and I'm very excited that we're starting to move towards being able to do it hundreds of and thousands of these with this Editron technology that I mentioned. But I'll show you a traditional genome editing experiment. So here are the predictions for one patient variant in a locus that contains several genes that are known to be important for heart development. And we have this gain of chromatin contacts predicted as a result of the deletion of a sequence in the middle there. When we collected actual chromatin capture data in the wild type, we see blue as predicted, so not much chromatin interactions there. And then in the cell line where we inserted the deletion, we see a gain of interactions exactly as the model had predicted. In fact, a bit stronger even than the model's predictions, which is something we've noticed with all of these sequence 2 activity models. They often sort of under-predict the actual effects of the variants, which is an interesting problem to be working on now. Besides this phenotype, we did RNA-seq, found differentially expressed genes in this locus, and actually hundreds of differentially expressed genes genome-wide. So my collaborators in Benoit Bruneau's lab are now investigating this further in terms of functional studies and cell lines and animal models. But this is a variant that would have been completely overlooked by any of the existing tools for prioritizing variants. And while we chose this locus because it had interesting cardiac genes in it, we see this same kind of pattern in loci not currently associated with the disease. So we think that there may be cases where the proteins are completely intolerant to genetic variation, but you could cause disease by this genome folding mechanism. So in the last little bit, I want to talk about gene regulation. I'm not going to go step by step and build it up quite as much as the last story, because I want to focus on some problems we encountered and the solutions that we developed. But what a sequence to expression model looks like is you could start with personal genomes, cases where you have whole genome sequencing and matched RNA-seq, like the GTEx cohort that we heard about. Build a sequence to RNA model, score non-coding variants that might affect expression of genes, develop candidate causal variants, and then go and engineer these into isogenic lines, like what I just showed for the genome folding variant. And our ambition here is to do this in a very high throughput with the Editron technology. This is all in theory feasible, but we encountered a problem, which others have also noticed, so including Sarah's lab, which is a lot of these deep learning models that perform very well at predicting gene expression when you want to ask whether the one gene's expression is predicted to be high compared to another gene's expression low. So gene by gene across the genome, they do very well. When you focus in on one gene and ask the models to predict the relative expression of the different individuals for that one locus, these models do not perform well. And it's not surprising. They were trained on the reference genome. They've never seen a genetic variant, and they haven't seen variability in very much variability in the expression of a given gene. Some variability because they've been trained across cell types and tissues, but not a lot of variability, and not the kind of variability that might be functioning at a population level here. So showing the informer model, but this is true for many sequence to expression models that are out there. Here's one gene, just to make it easy to understand, BTNL3. The horizontal axis is the variability in expression across individuals. Here from the GTEx cohort, these are held out individuals that weren't used for model training, and there's a dot for each individual. You can see a wide range of observed expression levels, and the model's just predicting about the same expression for everybody, no variability. So there's a flat line, no correlation between observed and expected, whereas a linear model, like an elastic net, does pretty well. And that model is not seeing the whole genome sequence. It's using only the genotyped sort of commonly variable sites. So just based on SNPs. And so we asked ourselves, well, maybe Informer just didn't get to see the data that ElasticNet has seen. What happens if we train a model like an Informer model, but on the task here of predicting personalized gene expression? And indeed, our model called Performer is able to correct this problem. So for this particular gene, you can see that it is performing even slightly better than the ElasticNet in terms of both the Pearson correlation coefficient and the coefficient of determination. Neither our model nor ElasticNet is perfect. There's still some things to explain here, but we've corrected this problem. And that holds across hundreds of genes. And so here I'm showing the distribution of differences between our model in blue or the informer model in green compared to an elastic net. So I'm taking the elastic net as a baseline and I'm saying, given elastic's net performance, are we a little better, like for BTNL3, or a little worse? And our model in blue has a distribution centered around 0, maybe skewed a little high. We might have done a little better than elastic net, but not a lot better. But certainly we've corrected the problem with informer. And not just at that level of these summary statistics for individual variants and their effects, informer has also been pointed out, and we quantified here, to when it's predicting the effect of a variant, to disagree generally with the elastic net, and to have differences in magnitude, but also in the upper left and lower right quadrant sign mismatches, meaning that the variant was viewed as important by both models, but the deep learning model got the direction of the effect wrong. And we trust the elastic net because it is based on just putting that genotype, that single allele into the model, and it's just a fold change. You can't get that wrong, basically. And so there's a lot of sign mismatches, and those were largely corrected with our tuning on personal genome sequences. Much higher concordance with the beta coefficients. So another way of putting this is that our model can learn eQTLs, basically. And it does that, however, without forgetting something very important, that the informer model, just like the Chrome BPNet that we heard about, pays attention to variants that disrupt known transcription factor binding motifs. Whereas a statistical model, like an elastic net, will just pick any variant out of an LD block that is statistically associated with expression of the gene. It doesn't use genomic context. It doesn't have any learned knowledge of regulatory grammar. And so we sort of think of our model performer as a best of both worlds, that it has these features, and I don't show it here, but also most of its high-scoring variants are in chromatin that's predicted to be regulatory elements, or splice sites. So it retains these functional features of the deep learning model, but has gained the ability to make personalized predictions. It's not perfect. In fact, just like other deep learning models, it should, in theory, be able to extrapolate to loci that it's never seen before, but we have not accomplished that yet. So that is ongoing work. But we are emboldened to start generating data, because the exact data that you need to build a better model would include being able to probe some of these hypotheses that the model's generating and find out when we're wrong and when we're right. And as has been highlighted throughout the day as well, we will focus on patient variants, but you could, of course, put in synthetically designed sequences to test the understanding of the model as well. So we have a 3-year goal focused on congenital heart defects, autism, and on cancers, skin and colon cancers. And we will be using this platform with these deep learning models, this high throughput genome editing, and a whole bunch of different single molecule as well as single cell and bulk genomic assays to build up the data that we need to build the next generation of hopefully even better sequence to X models. So to end the day, I think this is maybe an appropriate slide. I think we're really entering a new era in genomics and in proteomics and in deep learning, working together, not just to interpret data and find patterns in data, but the models are getting good enough and smart enough and learning enough that we can start to use them to design experiments and to build the next generation of research technologies based on model predictions. And I'm super excited what the next sort of 5 or 10 years is going to hold with this design, build, test framework. So I'll stop there, thank folks in my lab and our funding sources, and I'm happy to take questions.